My name is Carolyn Schutt. I'm a bioengineering graduate student in the lab of Professor Schutt, and we're working on developing a new medical imaging technology that will allow us to get optical information from deeper within biological tissue. So this project is really exciting to me as a bioengineering student because it has a very direct application to the diagnosis of cancer. But also it's in the nanoengineering field because what we're doing is manipulating the nanostructure of these bubbles in order to get the kind of signal, in order to get a specific signal that we're looking for. And in addition, my advisor, Professor Schutt, is also a member of both the electrical engineering and the nanoengineering departments. So there's a lot of different expertise and disciplines coming together here to create this kind of technology. So currently there's a very high false positive rate associated with conventional X-ray mammography, and this is often thought to be because X-ray mammography is only looking at density differences in the tissue. So you could have a more dense spot of tissue, but it's not necessarily a tumor. So there's a very high false positive rate. So this leads to many unnecessary biopsies yielding benign lesions of people who don't actually have cancer. So if you use our probe, which transduces ultrasound energy into an optical fluorescence signal and is able to give you chemical signatures of the tumor, you could potentially improve the diagnostic accuracy of mammography because now you have more information about the tumor rather than just a density difference. You can see, oh, it's acting at a higher metabolic rate, there's oxygen depletion, there's angiogenesis, there's more blood vessels. So with this kind of screening technology, we could potentially have more accurate early detection of breast cancer. So this is a schematic of the ultrasound modulated fluorescence contrast agent that we've developed. It consists of a gas microbubble that is coated in lipids, which form a structure that's similar to a cell membrane. And inside of these lipids, we've placed a dye that will quench or emit less fluorescence when these dyes are close together. So when this microbubble is exposed to focused ultrasound, it will undergo size oscillations, expanding and contracting this outer surface shell. When the microbubble is contracted, the dye will emit less fluorescence. It's quenched. When the microbubble is expanded, it will emit more fluorescence. And what this does is it results in a fluorescence signal that is modulating here at a characteristic frequency, the same frequency of the applied ultrasound. This is very important for being able to pull out the signal from deep within the body. And this is one of the major problems that scientists have experienced in the past with being able to do optical imaging deeper within the body. So we think now we have a way to be able to pull out optical information which can contain important chemical information from deeper within biological tissue. So beyond my research here at UCSD, I'm also involved in a number of programs, one of which is the Gordon Scholars Program for Engineering Leadership. And through that program, I've been exposed to many different leaders in engineering fields and have learned a lot about how to present myself and also what it takes to be a true leader in the field of engineering. In addition to that, I'm also involved in multiple outreach experiences. I'm the outreach chair of the Bioengineering Graduate Society, and I particularly enjoy finding connections with the general public, especially young people, and trying to get them excited about engineering technology and hopefully influence them to go into science, technology, engineering, and medicine fields.